So then there's some other organelles that I want to be sure we touch on. The mitochondria, its job is to carry out cellular respiration, producing energy for the cell. And note, it has a large surface area on this inner membrane here. And that's significant. We're going to talk more about that in a later unit. So let's draw that one in. Next we have the mitochondria. And the mitochondria kind of has this sort of a shape to it. And the mitochondria is a pretty important structure, and we're going to be looking at it in a lot of detail later. So I think it's worth taking a moment to sort of dive in a little bit more and look at how that works. So let's do that. And so in this drawing, what we see is, a more detailed look at the different parts. So let's pause and again draw the entire mitochondria, labeling all the different parts of that mitochondria. And again, this becomes important in a later unit. So here's a larger picture of the mitochondria. And there's several structures that make up the mitochondria. So this sort of outer layer is called the outer membrane. Makes sense. This inner layer is called the inner membrane, which also makes sense. And the space between them is called the intermembrane space. So makes sense, right? Outer membrane, intermembrane space. Now you'll notice that that inner membrane is highly folded, right? There's all these little folds. So each of these folds is called, these are called the cristae. And the cristae's job is to increase the surface area for reactions that we're going to talk about at a later unit. The final structure I want to highlight here is the matrix. So the interior of that mitochondria is the matrix. I want to also point out that there is DNA in there. There'd be a single circular strand of DNA, but I'm just going to leave that out of this picture. The next structure is a chloroplast. Chloroplasts carry out photosynthesis, which produces both energy and sugars for the cell. These are only found in plant cells and some protists, so you don't actually have these. And so just like before, let's kind of pause. We'll kind of draw out a chloroplast and label all those different parts. And again, this all becomes really important in a later unit when we look at how photosynthesis works. So let's pause and do that. So now let's zoom in and look at the chloroplast. The chloroplast has this kind of gumdrop looking shape. And just like the mitochondria, there's an outer membrane and inner membrane with an intermembrane space in between them. Now, the interior of this is called the stroma, and the stroma is analogous to the matrix. Now, in the mitochondria, we had those cristae, those little folds that helped increase the surface area. In the chloroplast, there's a different system. They have these little stacks of individual discs. And each of these discs, each one of them is called a thylakoid. And the entire collection, like the whole stack of discs, those are called grana, or 1 granum. So basically, the purpose of these is, again, to increase the surface area for the reactions of photosynthesis, and we'll be looking more closely at that later. And then we have the cytoskeleton. The cytoskeleton, just like the skeleton of your body, kind of provides support and shape. That's what the cytoskeleton does for the cell. Cyto actually means cell, so cytoskeleton. It's scaffolding, so it supports the shape of the cell and anchors the organelle. And you can just kind of see it's like these little protein filaments running all over the place. I don't like to draw these into my diagrams because they would just turn into lines all over everything else and kind of ruin it. Note that bacteria don't actually have a cytoskeleton because they're so small. So there's actually different proteins that make up the cytoskeleton. Microtubules are little roads that vesicles travel on. This video is well worth taking the time to click. It basically shows this vesicle and this little kind of motor protein that kind of walks along the microtubule. And that kind of shows how the vesicle is able to move throughout the cell. So you can think of these microtubules as little roads that the vesicles travel on. They're also used for the movement of cells. So you're probably familiar with the flagella of a sperm cell, this like whip-like tail that powers it through. And then other cells have what are called cilia, which are these little hair-like structures that surround it. Um, and then microtubules also help in cell division, making up something called the spindle. And that's something that will come up later on in this course as well. So microtubules do a lot of things. Microfilaments are really just used in one place. They're used in muscle contraction. And so, something called actin and myosin. If you take human anatomy, we dive into that in a lot of detail. And then finally, intermediate filaments, they provide shape and support for the cell. So again, this is kind of that scaffolding that fills that entire cell up. So the last structure I want to point out is not entirely in the cell, actually. It's called the extracellular matrix. That is the proteins and sugars that kind of stick out of the cells. So for example, if you think of your face, you can look at the little picture in the bottom corner of your screen here. Your face, your skin is held on to the underlying muscle by connective tissue, and there's a lot of this protein called collagen in there. And so you can see that there's proteins in the cell membrane that attach to that collagen and kind of hold that cell in place. And so all of these little proteins out here, there's sugar groups and all sorts of things, that's all called the extracellular matrix. It's that stuff that sticks out of the cells. This video is well worth clicking and watching. It's very cool, and it highlights the importance of the extracellular matrix. It's just like a 2-minute clip about some U of M researchers who basically created a functioning, beating heart. And what they did was they started with a rat heart, and they used a solution to basically wash away and kill off all the cells that were in that rat heart. And what was left were these proteins that make up the extracellular matrix. And then they added stem cells to the mix, and stem cells have the capability of kind of turning into any other type of cell. And so, those stem cells they were developing somehow, and I don't think anybody really knows how they did it, but they did it. Using these little proteins, they were able to kind of figure out where they belong and what they should turn into, and basically reassemble themselves into a fully functioning heart that beat and everything. So potentially, we could use this technique to start with pig hearts and, add human stem cells and sort of create a fully functioning human, well, not really human, kind of human heart that could be used for patients who need a heart transplant. So very cool, kind of highlighting the importance of that extracellular matrix. So at this point, hopefully this picture that we started with doesn't look so daunting anymore. You can see the nucleus with the nuclear envelope and the nuclear pores. You can see the rough endoplasmic reticulum in blue here. In yellow, you can see these little dots. Those are ribosomes. You can see the Golgi apparatus, our little flattened pancakes over here. You've got the smooth ER that's blue but doesn't have the ribosomes attached to it. The mitochondria are here. Here you can see these proteins represent part of the cytoskeleton. So all those structures we talked about are here, lysosomes, etc. The only final thing I want to point out is just to remind us that this is, again, a very generic cell. And, keep in mind that there's lots of different cell types. So cells that are involved in digestion might have a lot of lysosomes in there, and cells that are involved in producing a lot of proteins might have a lot of ribosomes in there. So different cells have different structures. Don't think that there's exactly one mitochondria, exactly one lysosome, exactly one vesicle, etc. in every cell. In reality, they have lots of these structures, and then cells vary by type. Okay, little checkpoint there on the cell structures. So the final thing then that I want to look at is just the difference between a plant and animal cell. So, and then a prokaryotic and eukaryotic cell a little bit more. So plant cells are similar to animal cells, but they have a few unique features that we got to talk about. First of all, they have a cell wall, and that cell wall is made of a carbohydrate that hopefully you remember from the last unit, cellulose. Their vacuole is different, and if you look at that picture here, you'll see that the vacuole is a single large central vacuole, versus in animal cells, they have lots and lots of these smaller vacuoles. And then finally, they have chloroplasts, represented by these green structures, and animal cells do not. Chloroplasts, remember, do photosynthesis. Prokaryotic cells, then, these are the cells, remember, that do not contain a nucleus, and they're different in a number of significant ways from eukaryotic cells. They contain only, this is it, the DNA, but it's in a circular shape. So in eukaryotic cells, we didn't really talk about this, but the DNA is arranged in chromosomes. In prokaryotes, it's a circular shape. They have a cell wall, but it's made of a different structure. It's made up of something called peptidoglycan instead of cellulose, so it's a different cell wall. They contain a plasma membrane, they have the cytoplasm, and they have ribosomes. Note that there are no nucleus or other organelles, and note that they are much smaller than eukaryotic cells. So keeping track of the difference between a prokaryotic and eukaryotic cell is very important. Keeping track of the difference between a plant and animal eukaryotic cell is also important. Final thing about cells. Here's kind of a weird, interesting little fun fact. Mitochondria and chloroplasts have some very interesting features. It turns out they have their own DNA. And that DNA is circular in shape, which is exactly how bacterial DNA is arranged. Mitochondria and chloroplasts are actually about the same size as bacteria. Mitochondria and chloroplasts have membrane lipids similar to the membrane lipids found in bacterial cell membranes. Mitochondria and chloroplasts, when the cells reproduce, these organelles will also make copies of themselves, and they do it by this process called binary fission, which is how bacteria divide. And then the mitochondria and chloroplasts aren't as tightly associated with that endomembrane system as other organelles are. So all of these observations have led scientists to an interesting conclusion, and it's called the endosymbiotic theory. And this theory is that mitochondria and chloroplasts are actually derived from bacterial cells that were engulfed in larger cells. So here you have some ancient ancestral cell, and you can see infolding of the cell membrane. Why would that make sense? Think about it. Why would that be advantageous? It's for surface area, right? So you get increased surface area so that you can do more chemical reactions on that membrane, and then eventually those branch off to become the organelles. And then at some point in the history of life on Earth, this little bacterial cell got pulled into that thing, and a little association was formed. Mitochondria, you may recall, is what produces energy by cellular respiration. So you can imagine that if this little bacteria was producing energy, this larger cell would be cool with that. Like, all right, stick around. And in exchange, the larger cell provides protection for the smallest one, or the smaller one. And so then a little association gets formed, and this is what eventually gave rise to modern eukaryotic cells. Another ancestor, maybe a second bacterial, got pulled in. This one could do photosynthesis. Great, that can make sugar and energy from sunlight. And this one gave rise and became a plant cell. So yeah, so this is kind of a fascinating idea because it means you have this little ancestral hitchhiker inside every single one of your cells. And this association became so tight that, now we don't even consider these different organisms. They're just kind of all one and the same. So cray cray. All right, so that is it for this unit on cells. Here would be a really good practice activity. You could do a Venn diagram or a double bubble or whatever you like to compare and contrast these two categories of cells and see how you do. Thanks.